Good afternoon, everyone, and welcome to Grand Rounds. My name is Tom Fardon, and I am the chair and organizer of Grand Rounds. Thank you very much for joining us today. Hopefully, you got my email that described the challenges we have with trying to get email lists updated, particularly with the NHS Tayside mail moving to nhs.scot. So we no longer have the same mailing list. So if you've joined this because you got the email, you don't have to do anything. But if you joining this because you found out by some mysterious way, then please do email me and let me know your email address. I will add you manually to the mailing list, and then you'll never miss out on my hugely entertaining emails. So hopefully we'll be able to broaden the net. And one of the groups of people who don't tend to come along is the students. I have encouraged them that these meetings are just as much for them as they are for anybody else, and hopefully they are interesting, entertaining, educational. So if you do have some students who are working with you at the moment, please do encourage them to sign in, or go to our YouTube channel, which you can find with the link in the email, or you can just Google or put into the YouTube Dundee Grand Rounds. Like, subscribe, hit the little bell, and you'll get notifications when new presentations are uploaded. I do it intermittently, but we are catching up. Speaking of medical school, I'm very pleased that this week at Grand Rounds, Professor Rory McCrimmon, who is the Dean of the medical school here in Dundee, has finally agreed, as I've been chasing him for a while, to speak about his research interest, which he's a professor of diabetes and metabolism. So his interest is in hypoglycemia, and his talk is entitled Hypoglycemia, Insulin's Dark Side. Now, if you can make sure your microphones are muted, apart from Rory, obviously. If you do have questions, please put them in the chat box, and I'll ask you the questions at the end. Or we might even tempt you to turn your video screen on so you can ask them in person. Rory, thank you very much for fitting us into your very busy schedule. I really appreciate you coming along, and I'm looking forward to your talk.
Thanks, Tom. It's really nice, actually, to be able to give a talk about research and not necessarily about the medical school or finances or some aspect of education for a change. And yeah, as ever, before I start one of these, again, just want to express my thanks. I know a huge number of you on the clinical side have made this huge, I see Jonathan O'Riordan there, one of the first, made that big transition towards teaching an online blended learning. Well, I know it's been a challenge, but I think it's been very impressive, and the feedback from the students has been excellent. So once again, thanks for all of your support at this time. So I'm going to talk about hypoglycemia, and this is most of my research program. So this is what I do in the other part of my life, if you like, outside of the deanery itself. And the title Insulin's Dark Side is a sort of homage, if you like, to a chap called Bob Sherwin, who was an American professor of diabetes that I worked with in Yale for about 10 years. And he was a really fabulous person, and was an incredible mentor to me, and helped develop a lot of those sort of research skills that I had, or what the few that I actually possess, I suspect. He tried his hardest to hone a researcher out of me, but probably struggled a little bit towards the end. And insulin, of course, it's an absolute time if you think about it, because next year is the 100th anniversary since the discovery of insulin. And there can be no doubt about the impact that insulin had on a disease, type 1 diabetes, that up until that point was effectively a death sentence. So insulin came along and it transformed people's lives. But very quickly, it became apparent that it was an imperfect therapy, as many therapies are. And the downside was hypoglycemia. And I'll talk a little bit now then about the sort of research that's gone into trying to understand that. But it's a medical conference, so not surprisingly, I'm going to start off with a couple of cases to illustrate the sort of people that come into a clinic I run for people who have problems with problematic hypoglycemia. And this is a 39-year-old female, type 1 diabetes of 21 years duration, on slightly old-fashioned, Humalog mixed 25, 28 units twice daily, HbA1c in the moderate range of 66 millimoles per mole. And if you look on the right, you can see what we increasingly use now, which is a continuous glucose monitoring profile. So this samples interstitial glucose every 15 minutes from underneath the skin, gives you a really good profile of what your glucose is throughout the day. And you can immediately see here on her profile that she's wildly fluctuating between high glucose and very low glucose. So the green bar here represents the normal physiological range. And you can see these repeated episodes of low glucose at various times through the day and often overnight. And she had developed what was called impaired awareness. So by that, I mean that she didn't recognize she was hypoglycemic until her blood glucose values fell down to 1.8 to 2.5 millimoles. That's very low. The lowest limit of normal, as you know, is 3.9 millimoles per mole. So she's not recognizing she's hypoglycemic until it's very low. And in fact, mostly her hypoglycemia was detected by a partner, or if she was walking around the room, or in the house, suddenly found herself in the kitchen, had no idea how she got there, she would take a blood glucose reading and recognize she was hypoglycemic. So a profound impact on her way of life. This is another profile, case study 2 here, a 19-year-old male student, type 1 diabetes, 8 years duration, on a pump, good control, HbA1c 58 millimoles per mole. But again, despite using pump therapy, you can see these repeated episodes of low glucose and high glucose. And he had impaired awareness of hypoglycemia to the extent that he had recurrent severe hypoglycemia, so where you've lost consciousness or require assistance to recover. And he had lost his driving license because of that. He was suffering from repeated seizures as a result of hypoglycemia. And he'd broken his collarbone following one of these hypoglycemic-induced seizures, or he'd been out clubbing and drinking on that night. And alcohol suppresses hepatic gluconeogenesis, particularly in the fasted state, and increases your risk of hypoglycemia. So you can see that the fairly dramatic effects this has on people's lives. And I'm reminded also of a case I had recently of a young woman who ran her control very, very high. She had an HbA1c that was over 100 millimoles per mole. She had significant complications with sight-threatening retinopathy and was showing proteinuria. And she'd only had diabetes for 18 years. And, of course, you're asking why her control is high. And she had fear of hypoglycemia. She had been on holiday in Bulgaria. She had stepped out to go for a walk and woke up 2 days later in hospital. And she said hypoglycemia was too close to being dead. So she just ran herself high all the time. And that fear of hypoglycemia prevents a lot of people achieving the sort of glycemic targets that they need to achieve if we want to reduce the risk of microvascular disease, sight-threatening retinopathy, end-stage renal disease, etc. Here I'm just going to show you a profile here. Katrina, my clinical research fellow, gladly submitted her own profile. And what you can see is the type 1, individual with type 1 that I've just shown you there in green with this massive fluctuating pattern. And then here is someone in yellow without diabetes. And you can see the vast majority of time, apart from, and I have to talk to her, a very unhealthy breakfast, most of her glucose levels are well within the normal range. And they rarely, rarely come outside of that. So hypoglycemia in people without diabetes is rare. You will see some episodes here where it looks like an individual is hypoglycemic, and I think there's more errors in the machine itself, although hypoglycemia does exist under some certain circumstances. This is me, you see. I'm just showing this to embarrass my clinical research fellow because I can see she's on the call, but I have a much healthier breakfast. That's what I want to say to Katrina. Porridge in the morning, Katrina, that's what you need. But again, you can see that in someone without diabetes, you've got a very, very flat profile over time. Okay, so what I'm going to talk to you about is the sort of work that I've done over the years, which is really looking at hypoglycemia and impaired awareness, a brief introduction of that. And then a little bit about the research that I've done in the lab around where and how we detect hypoglycemia and why this system becomes abnormal over time, leading to a process that we've recently proposed called habituation. So hypoglycemic reaction, I told you insulin was discovered in 1921. The anniversary is next year. And here in 1923, we're already describing the so-called hypoglycemic reaction. And I think all of you are clinical and most of you will recognize the classical symptoms of hypoglycemia and how it presents individuals. What also became rapidly important was this changed over time. So here are descriptions in 1927 and from R.D. Lawrence, part of the discovery of insulin in 1941, showing how reactions over time differed so much from the original ones that patients become dangerously unaware of the onset. This is impaired awareness, so a reduced perception of the development of hypoglycemia. It's quite common. It's around about 25% of all people with type 1 diabetes in most surveys. Same in kids, actually. In children and adolescents, about 25%. And actually, increasingly, we now see in adults with type 2 diabetes, with longer duration type 2, who are on insulin therapy. So a quite common presentation. And the main problem with impaired awareness of hypoglycemia is that it markedly increases your risk between 3 to 6-fold of severe hypoglycemia. And that is, again, when you lose consciousness and require external assistance or you're hospitalized. And so here what we have in the orange bars is the frequency of severe hypoglycemia, those with normal awareness versus those in blue here with impaired awareness, and it's far higher. And this work was from Scotland. It was done by Anne Gold when she was working in Edinburgh a number of years ago, and this has been repeated in a number of studies. So physiologically, what is happening? So I'm going to show you some, just going to illustrate that from some work at a gross level, if you like, from Stephanie Amiel, done many, many years ago when she was also working at Yale University. Stephanie is chair of diabetes in King's College in London. And she took individuals and gave them a constant low-dose insulin infusion. Now, if you look at those with conventional therapy, now at the time, way back in the sort of end of the 1980s, conventional therapy in America was once or twice daily insulin. And those people had really quite poor glycemic control. So if you gave them a constant insulin infusion, you'd see that the glucose levels here in these orange dots would drop, and then it would plateau around about 3.5 millimoles per mole. It wouldn't come back up again. It would just sit there with this constant infusion. This is a balance between the glucose-lowering effect of insulin and what is called the counter-regulatory response, hormones glucagon, adrenaline, noradrenaline, cortisol, growth hormone. Now, she took people then who'd been intensively treated. So this was multi-injection therapy with insulin or the insulin pump. HbA1c, much tighter control now, down to sort of 7% or 53 millimoles per mole in the old money. And you gave them exactly the same insulin infusion, you matched your population, and their glucose levels fell far further, down to 2.5 millimoles per mole, and plateaued at a much, much lower level. Now, this physiological response you see in glucose was matched by a lower response in terms of hormones and also of symptoms as well. So there was an altered thresholds or activation. If I were to take someone without diabetes and give them the same insulin infusion, their glucose levels would drop, but then they would counter-regulate strongly, and the glucose levels would come up to the normal range, because that's part of the normal homeostatic response. Done a lot of research over the years, and there are two major reasons. So this first defect is because of the failure to shut off insulin. So if someone without diabetes becomes hypoglycemic, they stop producing insulin. That would be a normal physiological response. So insulin levels fall. But if you've taken an injection of insulin, it's sitting under your skin or being delivered by pump, you can't switch it off. It continues to act and continues to deliver glucose down. They also have this rather fascinating defect in alpha cell glucagon secretion. So it's present in about 100% of people with type 1 diabetes after 5 years disease duration. The glucagon is present in the islets. You can see vesicles, you can see glucagon production. It's released to other stimuli, such as, say, arginine, but not to hypoglycemia, very specifically. The second defect results from recurrent exposure to low glucose. So the difference between the conventionally treated and the intensively treated one, we now know after a lot of studies, relates to recurrent hypoglycemia itself, a vicious cycle whereby hypoglycemia begets hypoglycemia. And we'll talk a bit later about why that might happen. So here's the net effect. If you drop glucose slowly in the individual without diabetes, you get a physiological response, you get warning symptoms, and then you get cognitive impairment. Whereas in the individual with type 1 and impaired awareness, you lost the glucagon response, you have a poor epinephrine or adrenaline response, you have reduced symptoms and reduced anxiety, a reduced behavioral response. So now the window between recognizing hypoglycemia and being absolutely unable to do anything about it because your glucose levels are so low is reduced markedly, and you get severe hypoglycemia. So where do we sense glucose? So that was the work. These were the physiological studies that had been done in the late 80s and 90s, and even I'm not that old, but I came in a little bit later on and started to look at some of the basic mechanisms. Now, we now recognize that most hypoglycemia is detected by the brain. There are specialized neurons, which I'll tell you a little bit later on, within the brain. So the brain is about 2% of body mass, but takes about 20% of the body's energy needs. And the brain, unlike most other organ systems, has no alternate fuel stores. We have a tiny amount of glycogen in the brain, nothing like the quantity in muscle and liver, and we have very little fat other than in the lipid membrane, which you wouldn't want to break down. So there is a need for glucose, and we've evolved multiple systems, therefore, to ensure that the brain gets that glucose throughout the day. We also now recognize that within the brain, there are discrete regions that appear to play a big role in sensing glucose. So here I'm showing a part of the hypothalamus with the ventromedial nucleus and the arcuate and the dorsomedial nucleus, and the medial, here is the medial amygdala. And what we're staining here using in situ hybridization in a rat brain is for glucokinase mRNA. So you remember your biochemistry, glucokinase is the first committed step in glucose metabolism before it enters oxidative phosphorylation. And these part of the brains have glucokinase mRNA. If you microinject, in an animal model, of course, 2-deoxyglucose, which is a non-metabolizable form of glucose and creates like a local hypoglycemia, you stimulate a counter-regulatory response. Alternatively, and in both of these regions, alternatively, if you put glucose into these areas but create whole body hypoglycemia, you suppress the response. So these are shown to be glucose sensing. And we now know that there are multiple areas in the brain where this occurs. So there's glucose sensing. This is a sagittal section through a rat brain, but effectively in hindbrain and in forebrain and hypothalamus, these areas are all glucose sensing. And the little red stars here is where people have used electrophysiology, and our own Mike Ashford was one of the first to demonstrate this, that these neurons detect and respond to changes in blood glucose. And they form an integrated network. There are peripheral sensors, and also they integrate it with other hormones. We know, for instance, that during anorexia, that women become anovulatory. And so these systems are all connected together in terms of integration of glucose homeostasis, energy metabolism, and other aspects such as fertility. So leptin and adiponectin and all these other hormones will affect glucose-sensing neurons, and they speak to each other on a regular basis, like any decent homeostatic system. So where do we sense glucose? This interconnected circuit, specialized neurons found in multiple brain regions and in the periphery. So the next question, of course, is how do we sense glucose within these neurons? So I'm now focusing in on a specific neuronal population. This is a candidate glucose sensing neuron population, hypothalamic hypogonadotrophic GT17 cells. It's work done by Craig Beal, who was working with Mike Ashford and myself a number of years ago. And here I'm going to show you, first of all, the beta cell. So we all recognize the beta cell as a glucose sensor. And the beta cell is actually the one organ system outside of the body that you can record from electrophysiologically. The beta cell is like a modified neuron in many respects. And it fires away at 10 millimolar glucose, and you drop it to 2 millimolar and it so-called hyperpolarizes. You add in glucose again and it fires. Now, if we take this neuronal population, it does the same. Now, you'll notice that the glucose levels are much lower, and that is because interstitial glucose in the brain is about 20% that of plasma. It's far, far, far lower, so there's much less capacity for change. But if you drop glucose levels to 0.5 millimoles per mol, cells hyperpolarize, and then they fire off again at 2.5, and you can show that there is a dose response. So here we have neurons that respond with a changing in firing rate depending on the blood glucose, so they're glucose sensitive. And Craig Beal went on to show that they were also regulated by glucokinase. So here we've added a glucokinase activator and show we can restore sensing, or we can mimic hypoglycemia with an ATP-sensitive potassium channel blocker, opener, such as diazoxide or a compound called NN414. And as an aside, here on the left, I'm showing you a picture of a fly brain. And in the fly brain, insulin and glucagon are neurotransmitters found within the brain itself. So there is a teleological role, perhaps, for insulin. In fact, there's a lot of research ongoing as to whether insulin actually is produced within the brain in humans as well. Now, we took that then step a little bit further, and we started to look at in vivo models. And the sort of studies you're seeing here involve something called microinjection, where we microinject in an animal model, an awake animal model that has been anesthetized and has had catheters surgically implanted targeting brain regions a week prior to study. And then we are able to clamp them with vascular catheters that are put into aorta and IVC. And here what you've got is a control study in black where you see this brisk response in terms of adrenaline to hypoglycemia. But in contrast, if we microinject glibenclamide, a potassium channel blocker, if you like, we suppress counterregulation. Conversely, on the other side here, if we microinject a specific or non-selective potassium channel opener, NN414 diazoxide, now in the orange, you're amplifying the counterregulatory response. So we've shown, obviously, in a cell that you can do this. Now we're showing in vivo in a rodent model that if you activate or suppress these neurons, you can drive or suppress counterregulation. So then we went on to look at humans, and this work was done by Priya George, our very own Priya George, who's now a consultant here in 9 Wells Hospital, and Priya was looking at people with long duration type 1 diabetes. They're brought into our clinical research center. They're undergoing what's called a clamp procedure. So here you've got your hand and a hand box which arterializes the blood, so it gives you an estimate of what the arterial glucose concentration is going to the brain. We're obviously infusing insulin and glucose to maintain glucose at fixed levels. We're taking lots of blood samples. We're doing cognitive and symptom tests. And what Priya showed was a significant increase, about 37%, in the counter-regulatory response to hypoglycemia with the oral agent diazoxide, which is an oral potassium channel opener that is used clinically. So again, this is one of the great joys of translation, when you can take something from cell to animal to human and to show an effect of your therapy, but also confirmed the fact that the K8P channel had played a crucial role in glucose sensing. Priya also went, and we're in a world of pharmacogenomics now, I've discovered, of course, in Dundee, and she was able to show that those people who had an E23K mutation of the SIR1 channel responded differently from those who didn't have a mutation. So a way of looking perhaps in the future where we might do more selective interventions in this cohort. Unfortunately, Novo Nordisk then pulled out of making small molecules, and that whole NN4 and 4 area fell flat in its face, and I need to figure out some way of reinventing it at a later date. So how do we sense glucose? Well, there are two basic populations of neurons: those who are activated by a high glucose, they're glucose excited, and those who are activated by a low glucose, and they are glucose inhibited. Both of them use these classical points of the glucose sensing: glucokinase, AMP activated protein kinase, K8P channels. The excited neurons appear to secrete the inhibitory neurotransmitter GABA, whereas the inhibited neurons secrete the excitatory neurotransmitter glutamate. And that's about as far as we've got in terms of sensing. There's some work on SGLT2s and some work on TRP channels within the brain itself. So recognizing that we have these regions of the brain that sense glucose, we've got these specialized neurons that sense glucose. Why does it become abnormal? Why over time, in the face of repeated hypoglycemia, do we get impaired awareness? So the, apologies, the classical study that started this off came from Simon Heller. I'm gonna have to take a drink of water. Simon Heller is a consultant physician from Sheffield who was working in St. Louis with a chap called Phil Cryer at the time. And what Simon did is we took people without diabetes into a lab, and they were studied in two separate occasions. On one occasion, they brought into the lab on day one and made hypoglycemic. I have to get James to test me. The following day, they were brought in again. And they did this, whereas on an alternate day, they had a euglycemia study on day one, that's normal glucose, and day two, they had hypoglycemia. So if this represents, if you like, your first response to hypoglycemia, and you see this very brisk adrenaline and symptomatic response, as I've showed you, where the group who had hypoglycemia the day before, when brought in for a second episode, now it was markedly suppressed. So it reduced adrenaline response and reduced hormones. And this was followed up by multiple studies in humans with and without type 1 diabetes and in various animal models, all showing exactly the same thing, is that the depth, duration, and frequency of hypoglycemia all determine the sort of size of defect that you see down the line. And Phil Cryer proposed that this was a maladaptive response, and he called this a rather cumbersome name: hypoglycemia-associated autonomic failure. And in the last few years, I've been working with certain of my colleagues in my group to show that this is wrong. Fundamentally, it is hypoglycemia-associated, but it is not an autonomic failure. It's more an adaptive system, and I'll show you why we think that might indeed be the case. Now, this is just to illustrate that there's been an awful lot of research in the background, a lot of which came from our group, but also from many other groups, would have looked to see, well, why might these neurons no longer be sensing glucose in the same way? So, for instance, you may simply be much better at transporting glucose. So you can get that glucose, you can suck it into the neuron, you can drive it through oxidative phosphorylation and maintain that K8P channel activity. Alternatively, can you use alternate fuels? There isn't much in the brain, but there was some evidence of what's called glycogen supercompensation. And a lot of people believe that neurons predominantly use lactate as a fuel. So could you be generating more lactate? Could you be changing the way you sense glucose through alterations in glucokinase, AMP kinase? Could you be affecting in some ways neurotransmitter release or structurally changing synaptic inputs in different parts of the brain? Might it in fact be a change with so-called gliotransmitters? So neurons are surrounded by glial cells who produce lots of hormones, inflammatory markers like cytokines, and might they also be affected it? Or could it be peripheral signals that have been worked on opiates, steroids, cytokines, leptin, and ghrelin? So multiple mechanisms. And as ever, my guess is going to be very, very complex and involve a lot of these together, and no single mechanism is likely to explain this. And it may be different parts of the brain are affected. Now, what we can do is use a lot of different models to address this question, because as I said to you before, whether you take the C. elegans worm, the fly, the mouse, the rat, the dog, or the human, they all respond in exactly the same way to recurrent hypoglycemia. So as an example of this, Lizzie Haythorne, who was doing a PhD with myself and Mike Ashford, Lizzie took this neuronal population I showed you earlier on, the GT17 cells, and as I showed you earlier on, when you drop the glucose, you make them hypoglycemic. Now, if you take those same cells in media and expose them to low glucose for 3 hours on 3 days, and then bring them into the lab and make them hypoglycemic again, well, now you have an unaware neuron. That neuron no longer responds to that glucose. It continues to fire normally. And we can use this as a means of trying to understand mechanism. So I won't go into too much detail here, and I'll flick through this very rapidly. But what Lizzie showed here was that glucose uptake using 2DG was not affected by recurrent hypoglycemia. But when she used something called a seahorse extra cellular analyzer to look at metabolism within the whole cell itself, what we actually showed looking at oxidative phosphorylation, OCAR, or glycolysis, ECAR, that effectively both of these were suppressed. And we went on to show that effectively glucose was being diverted down the pentose phosphate pathway rather than necessarily being used as a fuel. So the cell, if you like, was becoming hypometabolic in response to low glucose. And we're continuing to look at that because, interestingly, people with type 1 diabetes exposed to recurrent hypoglycemia gain weight. So there may be an explanation for that. Now, of course, if your cell is no longer using that glucose to generate ATP, well, why is it continuing to fire? And again, I'll just show this very, very briefly. I'll flick through it quickly. But what Lizzie did was show that these K-ATP channels had been desensitized after recurrent hypoglycemia. So they are opened by ADP. And as you drop ADP and add in ATP and add in ADP, what you can see is this whole threshold for activation has now changed. There's a single identified single K-ATP channels, and showing that their sensitivity to ATP has now changed profoundly. And that explains the disconnect between the metabolic state and the signal that's happening later on. The mechanism for that, we don't know. Is there a model for this in nature? Well, there is. The bears. Hibernation is the classical model for this. Bears become hypometabolic during periods of prolonged fuel deprivation, and they survive for a long period, of course, because of that. And then they store up their fat and their energy stores when they start to feed again during the year, hibernate again over winter. So hypoglycemia and hypometabolism and hibernation, I think, is a useful model to look at it in that context. So why do I think, therefore, it's not autonomic failure? Because what I think we've demonstrated is a system that is adapting. And what I think, and I'm just illustrating some of the data here, but effectively, if you consider that in normal and abnormal, in people without diabetes, when glucose levels fall, they recruit more and more of these glucose sensing neurons until you have an optimal glucose counter-regulated response. Well, in type 1 diabetes after recurrent hypoglycemia, you're just seeing a left shift in this activation threshold. Whereas we know in type 2 diabetes, you see a right shift. So often I'm asked, for instance, because they're exposed to chronic levels of high glucose. So I'm asked, can an individual be hypoglycemic if their glucose level is 5 or 6? Well, actually, physiologically, you can be. You're not biochemically hypoglycemic, but if poorly controlled type 2 diabetes, you can absolutely be physiological hypoglycemic at this level. And that is a classical sensory motor response in a way. So I'm going to go on. I'm going to take a real lateral shift for you and show you the next in my sort of David Attenborough view of research and science. And this is called the sea slug, or Californica aplasia. And the reason I'm showing this is it's a model for something called habituation. So if you take the sea slug and you flip it over, there's great videos you can see on YouTube for this. And you take a little stick and you tap it, it's what's called its mantle. You get a classic withdrawal reflex. But the more often you tap it, eventually it stops responding, and that is called habituation. So why am I showing you this? Well, habituation is the reduction of psychological, behavioral, or physiological responses to a stimulus, a homotypic stimulus, as a result of repeated or prolonged exposure. Well, hypoglycemia, in recurrent hypoglycemia, we the same thing. We see a reduction of psychological, reduced anxiety, behavioral. Why is it someone with recurrent hypoglycemia can do a blood test to show they're hypoglycemic? can be given a little device to show that they're cognitively impaired, but still think they can drive for the next hour before they need to take any food. So the behavior is abnormal. And the physiological responses I've just showed you is abnormal. So this fitted with the idea of habituation. The other reason I think that it fits with habituation is in essence, actually, although it's used extensively in psychological research to talk about behavior, it is in essence a sensory motor reflex, the gill withdrawal reflex. In many ways, the detection of hypoglycemia is, if you like, an internal sensory motor reflex. You internally sense glucose, and your motor response is counter-regulation. And therefore, you have these glucose sensing elements, you have premotor control, you have an autonomic and neuroendocrine motor response, and you have these integrative networks in hindbrain, hypothalamus, and limbus. And that's fairly classical for a lot of systems that work within the brain itself. So we looked at this. So there are 10 criteria for habituation, one of which is that if you give a response, repeated applications of that stimulus lead to reduced response. Well, I've just shown you with Simon Heller's work, acute hypo and recurrent hypoglycemia leads to suppressed counterregulation. If the stimulus is withheld, the response recovers over time. And we know with strict avoidance of hypoglycemia, so these are individuals before and after strict avoidance, leads to at least a partial recovery of the adrenaline and symptomatic response to hypoglycemia. So hypoglycemia avoidance works. The next thing we tried was something called dishabituation. And this is work done by Alison McNeely, who's been in my lab and working with me now as a colleague for a very long time. And this is where if you present a different or novel stimulus, you can recover the habituated response. So what Alison did was she took a rodent model in this case. They were exposed to recurrent hypoglycemia over 4 weeks. And then, so we have the control animals, acute hypoglycemia. We have the recurrent hypoglycemic animals. And then in this group here in green, after the last hypoglycemic episode, 24 hours later, they've undergone a high intensity exercise program. We put them on a treadmill and we run them really hard. And then the following day, 24 hours later, they have this hypoglycemic clamp. So we match their glucose levels. And I'm going to just really get you to focus on this graph down here. You can see the brisk adrenaline response in your control group. You can see the classical suppression after recurrent hypoglycemia. And now after a single burst of high intensity exercise, you've restored it. So that would be consistent with dishabituation of a habituated response. Alison then went to show using proteomics, a SILAC mouse model and proteomics, to show that this appeared to be potentially restoring glutamate excitatory activity within the hypothalamus itself. And I won't go into this slide in detail, you'll be delighted to know. Kieran, who is a medical student attached to our lab for a while, actually looked at the impact of cold exposure as opposed to high intensity exercise. And this was exposing a rodent model again to 4.5 hours at 4 degrees centigrade, but doing exactly the same type of trial with the recurrent hypoglycemia over 4 weeks, the cold exposure, and the following up hypoglycemic clamp. And Kieran, if I just take you down to this graph here, did some really interesting work, which he's now been able to submit for publication. So great for medical students, showing control study in blue, suppression with recurrent hypoglycemia in green, and then the recurrent hypo plus this cold exposure 24 hours later showed now restored the adrenaline response to hypoglycemia. That gives us another potential therapeutic tool. Because of course what we need to do is we're showing this as an animal model. And again, it's my lab, it's a translational lab. Can we show this in humans? So this is Katrina Farrell. Katrina's doing a PhD with me at the moment. And in the first study that she had, which she published last year in Diabetologia, she now took individuals with type 1 diabetes, long duration, with impaired awareness of hypoglycemia, and compared a rest, a control study day, with a clamp following a high-intensity protocol, which was basically 30 seconds on, 2 minutes off, repeated 4 times. This is me showing that I can still exercise without dropping dead on an exercise bike down in the CRC unit. So we brought in people with type 1 diabetes to do that, and then they had a matched hypoglycemic clamp in a crossover study. And what you can see here is in the blue are the group who had the high intensity program. You see this amplification of the adrenaline response the following day, improved symptom score, and the expected changes in cognition. So again, we've shown in an animal model in humans, the principle of dishabituation, which of course fits with the hypothesis that we are seeing a habituated response over time. And Katrina is now running a 4-week intervention trial called HIT for Hypos. And I think we're really, hopefully, who knows whether we can complete this trial with COVID, but if we can, I think it'd be really interesting to see the outcome of that. And we can then take that to a larger clinical trial. So that's hypoglycemia in some ways from the naturalist perspective, and it shows how I think there is a place still for using models to help inform on human research and to take us through to that experimental medicine place before we then move into the clinical trial view. And that, of course, is our great national hero, David Attenborough. So in summary of this very part, hypoglycemia sensing occurs, we now recognize, in this integrated network with mechanisms that are very similar to pancreatic beta and alpha cells. And recurrent hypoglycemia, I think, initiates this adaptive response in the brain that increases the threshold for counter-regulatory hormone and symptom responses and impacts on cognition. We think this develops through a process we called habituation. It's been around for years, discovered in the 1940s. And that we might be able to use novel stimuli such as high-intensity exercise or cold exposure to reset the system for people with type 1 diabetes, which may work through restoring these glucose-sensing pathways in the brain, or may not. It still has to be asked. But the story slightly continues here, and I'm going to add a little bit more to it, because if this is an adaptive mechanism, why does it lead to severe hypoglycemia? And why does it appear to be associated, hypoglycemia, with increased morbidity and mortality in diabetes? And this is some of the work that's going on in the lab at the moment. So I know I'm not going to talk for too long. I already have talked for long enough. So I'll show it very quickly. So the first is that although habituation, which is a form of adaptive memory, is a physiological response, as I alluded to in those first few slides, hypoglycemia in type 1 diabetes occurs in an unphysiological context, high insulin and absent glucagon. So no matter how well you adapt, if your glucose level is low enough, you're going to develop severe hypoglycemia with marked cognitive impairment. So I think that is why in type 1 diabetes, recurrent hypoglycemia, to some extent, it is a maladaptive response because it initiates this severe hypoglycemia, or this risk of severe hypoglycemia. The other thing is that why is it associated with increased morbidity and mortality? Well, I think here the issue is that we mostly study hypoglycemia. Here's another profile of someone who was hypoglycemic almost every night. But what you will also note is that after they've been hypoglycemic, they become quite markedly hyperglycemic. People with type 1 diabetes often don't correct to a normal physiological range. They correct to a hyperglycemic range. And people feel miserable, they feel terrible, so they take their iron brew or their Mars bar or whatever, and that drives glucose levels up very, very high. Now that is potentially a very marked cellular stressor, a little bit like post-ischemic reperfusion that you see in the brain. And so Alison again did a study where she took some animal models, type 1 diabetes models, and exposed non-diabetic or type 1 diabetic rodents to recurrent hypoglycemia, and did something called the NOR task, which is assessing memory. And what she found is that long-term memory wasn't particularly affected by recurrent hypoglycemia. And in fact, other research has shown it's enhanced after recurrent hypoglycemia in the non-diabetic model. But in the type 1 model, you saw both an impact of diabetes and a really marked effect of type 1 diabetes plus recurrent hypoglycemia to negatively impact on performance. She then started to tease apart some of the detail by doing this really complex clamp studies in mouse models here, where you've got we're able to manipulate the glucose in lots of different ways, either the glucose leading up to the hypo or the glucose levels on recovery from the hypoglycemia itself. So it's a massively complex study, but suffice to say that what she showed was that in response to hypoglycemia in type 1 diabetes, you saw this perhaps expected upregulation of beta oxidation and pentose phosphate pathway, which is helping you improve your capacity to degrade damaged proteins, but you show a reduction in stress responses in the so-called chaperone pathway. So your ability to deal with misfolded and damaged protein is impaired, and that leads to inflammation. And so what we think is happening in this context is that diabetes per se, poorly controlled diabetes, is a chronic inflammatory state and depletes your overall antioxidant response. Hypoglycemia is an oxidative stressor, but recovery is a profound oxidative stressor. And in those with poorly controlled type 1 diabetes, I think this ultimately leads to cellular damage and long-term consequences. And the work we are doing now, we're looking particularly from a cardiovascular perspective, because there's a lot of interest in that. So working with Faisal Khan here in the medical school and Callum Forteith, who's leading on this, we're using laser Doppler to look at microvascular function in mice using this same model itself. And I think hopefully we'll then go on to be able to look at humans. And I'll skip on this one, we're using flow loops to look at cardiac dynamics, physiological parameters such as end diastolic dysfunction in these animal models to see what is the impact of cardiac function. Of course, we can look at the heart tissue itself to look for fibrosis and change. So what are the therapeutic implications of this, just to round this all up? Well, hypoglycemia avoidance remains the mainstay of therapy, and that's through use of modern insulins, it's through use of education, it's through the use where appropriate of insulin pumps, continuous glucose monitoring, potentially cognitive behavioral therapies, and ultimately pancreas transplantation, although I think it's rare to have to get to this stage. Future strategies, you've probably all heard of the Artificial Pancreas Project, where we have pumps that are linked up with continuous sensors that give a feedback loop to control and regulate insulin secretion. It's really exciting areas in type 1 diabetes at the moment. Combination pumps with insulin and glucagon. Non-insulin adjunct therapies. So these are therapies like GLP-1 receptor agonists or NRF2 activators, which may help protect against damage. And then again, more exploratory areas such as dishabituation and different physiological stimuli to reset this homeostatic system. And these are areas that we're experimenting with here in Dundee. So what do we do for the first case? Well, the first case was pretty straightforward, and Katrina put him onto Degludec, which is a new basal insulin, and what you can see is, or put her onto Degludec, and you can see the significant improvement in her glucose profiles. But interestingly, a number of months later, she still hasn't restored her symptomatic and cognitive awareness, and it takes a long time to reverse. Evidence from pancreas transplant studies, means it takes 18 months or more of normal glucose levels for your awareness to be restored in type 1 diabetes. So it's a process that we need to work on. So these are all the people in the lab and collaborators and funders over the years that I'm very grateful to. I particularly want to thank Alison and Jennifer, who've been with me for a long time since I came to Dundee 10 years ago, and Katrina, who's been doing all of the human studies that I presented today. And that, I think, is me, Tom. Thanks for listening, folks.
Wow, that's science, huh?
That's what I do the rest of my time.
It's, if the students are watching this later on YouTube, you can see that you don't get to the Dean of Medical School for nothing. And I think this is the culmination of decades of research and being at the top of the field in this area. It's absolutely fascinating stuff. And it's nice to see the work of the postdocs. I think they don't get quite the recognition that perhaps they deserve in some instances, but you've acknowledged their work very well there. Does anybody have any questions? There's nothing in the chat, but have anyone got any questions they'd like to put to Rory just now?
We should acknowledge the medical student as well, Tom. I think Kieran Vignason did an amazing job, and I think it's really good to see that. So when there is that drive and willingness there, combining your DCAT vacation scholarship with your doing a BMSc, you did a tremendous project. Really gets a paper into Diabetologia, which is a really high impact journal.
Absolutely, and I think that's one of the things that we're really strong with here, aren't we? That
yes, absolutely.
Program, you can, if you're interested, there are people that you can get involved with, and there's a wealth of talent and expertise in the medical school and life sciences that we perhaps don't really know about. So there's two questions. We'll go with, I'm sorry, I want to have a C. Henstridge, so I'm going to go with C. Henstridge first.
All right, Chris.
And then we'll go on to Adinkova Kostova.
Oh, Albania, yeah. Okay, well, Chris, make it easy. I know you're a proper neuroscientist.
Well, we'll soon find out. Thanks, Tom. So my question, Rory, it's kind of I don't know if it's really a question or a comment, but I like this idea of the sort of hypoglycemia being like a habituation response, and the fact that you can kind of shock them out of it almost with high intensity training, or in particular that cold shock that you mentioned.
Yep. And
it kind of brought to my memory or attention some work by professor in Cambridge called Giovanna Malucci, who has done quite a lot of work into this idea of cold shock treatment and how it changes synaptic wiring and synaptic function.
Yeah.
And so would tie in maybe quite nicely with this idea of a sort of habituation-like response. And she's narrowed it down all the way to an individual molecule, essentially, and how important that molecule is for regulating that synaptic function. So I was wondering if there's any chance or opportunity you can maybe have a look in some of your models to see if these molecules are changing and maybe playing a role in that type of kind of habituation-like response.
Yeah, I think that would fantastic. I mean, we've just taken proteomics, Chris. So and that, as you know, is can be a bit of a snapshot where you just end up picking your protein of choice. So I think that would be fantastic. We'd love to look at that. I'm sure, Alison and I could, if we get some of those papers, that we can pull them out and have a look. I think it's really intriguing. We don't know, you see, whether when you, and from what I can see in the literature, people don't understand whether dishabituation is a true restoration of the habituated response or just a novel resetting of the system. But in the background, the habituated response continues. And that's why Katrina is doing this 4-week intervention trial with high intensity exercise to see whether, and of course you can habituate to the dishabituating stimulus. So how do we do this? I'm looking forward to, Tom, I'm really looking forward to doing a cold or a heat exposure trial in humans. I reckon a little portable sauna out of the back of the CRC. We can perhaps use ourselves as well as getting some people with type 1 diabetes into use, and then do that would be a great trial.
Okay, thanks, George.
Thanks, Chris.
I think I might be more likely to sign up for your HIT trial than the.
Yeah, well, we couldn't recruit you. You're too fit. It has to be a novel stimulus, I'm afraid. No.
Now then, one of your colleagues had a question.
Albania.
Hi, Rory. Yeah, this was great. My question is sort of following up on maybe Chris' comment that he just made. Do you think that the cold exposure and the high intensity exercise, their beneficial effects are mediated through a common mechanism, or do you envision different?
Really good question, Albania. So first of all, before I respond, everybody online should notice that Albania has just been awarded a big prize, one of the most highly cited researchers in the world. So hey, bow to Albania. That's an incredible achievement, I think, and shows just the incredible influence you have in the field, Albania. Now, is it a common mechanism? We are taking the two models and to try and triangulate and look at the hypothalamus to see whether there is a common mechanism, both in terms of doing some immunohistochemistry and looking at the brain, and also in terms of proteomics. So it could be, absolutely. You would assume that in these integrative centers, that's what you're seeing. Now, alternatively, as Chris has alluded to, if you change synaptic inputs, it could be that because there are both specific aspects of any homeostatic system and there are shared aspects. So it could simply be a different synaptic input from that system that happens to move into all of your sort of stress hardware, if you like, or it could be shared. Don't know. And I really don't know. And I've not been able to see anybody who's got the answer to it.
Thank you.
Thanks.
Tom, can I ask a question?
Go on then.
Rory, first of all, thank you. It's wonderful to have all that put together and see it all. I think that's the first time I've seen it all in one place, and it's really, really fascinating. So thank you very much indeed. I just have a question in relation. Fascinating to see how central the brain is to the regulation of this. What are your thoughts? There's a literature that suggests that recurrent hypoglycemia in patients with diabetes is responsible for dementia, the development of dementia in patients with diabetes. And how much do you think, or what do you think is the main driver of that? And is it some of your interventions, do you think there's potential there for prevention of the consequences of long-term hypoglycemia on the brain in terms of dementia prevention?
So, I mean, it really, again, Alex, great question, and a really complex one, as you know. And the problem here, of course, is that people who have dementia are at a high risk of hypoglycemia itself. So what is the chicken, what is the egg, right? Is it the dementia that increases your risk of hypoglycemia, or does hypoglycemia accelerate the process? My feeling is that hypoglycemia, given what I showed you in terms of the glucose variability, is actually accelerating the underlying inflammatory process. So it on of itself is not causing it, but it is both, it's adding to that chronic inflammatory process that results from hyperglycemia. Okay, now, can we do anything about it? Well, what I hope to do is working with, in fact, with Albana and others, is to look whether agonists of NRF2, which is this transcription factor that controls the antioxidant response, such as sulforaphane, or the great found in broccoli, and various other agents might actually help in this. The difficulty, of course, is how do you do the study? Because it takes 10 years to show some of these cognitive changes. And as you know yourself, it's one of the real challenges of dementia research is you've got to take people, I guess, with mild cognitive impairment and try and see whether you follow them up. You can do shorter term studies looking at, say, vascular function, but to look at cognition over time is hard. But again, I think there's certainly want to start looking in that space.
Okay. Great stuff. Anybody else got any questions? The clock is ticking on to 2 o'clock, and I need to put my PPE back on and get back to the COVID high dependency. So I'd just like to thank you again, Rory, for finding the time to come and speak to us about your research of interest. You mentioned how fantastic our DCAT program is, and next week, I got this wrong last week, so apologies to Ewan, who had a small acute coronary syndrome when he thought he was speaking this week, but he's definitely speaking next week. So Ewan Pearson is going to chair the session where he's going to bring along clinician scientists of 3 different levels: a foundation, an academic foundation doctor, a registrar in research training, and one of our consultant colleagues to speak about their experiences in research, present their findings, and give some insight for those of you, anyone in the audience who wants to be more interested in research and pursue that career. So it'll be a good session. The DCAT annual lecture is always an excellent session. I think this is going to be different from what we expect and what we normally have, but it will be no less excellent. So thank you, Rory. And I'll see. This will get uploaded by the end of the day, and I will see you all next week. Thanks very much.
Cheers. Bye.